Okay, we're going to talk about Chapter 19 on viruses. Viruses, by definition, are not alive because they don't consist of cells. They don't have a membrane. They do not have a metabolism. They can't reproduce on their own. They don't grow and develop, but they do have a big impact on things like this bacterial cell that's being infected by these viruses. First one discovered was tobacco mosaic virus, and it causes this mottled appearance of the leaves and stunts their growth. But when an extract from one of these infected plants was run through a filter that would normally remove bacteria, what the liquid that went through the filter when it was applied to a healthy plant would cause an infection. So it appeared this disease was caused by something even smaller and simpler than a bacterium. And as we know now, it turned out to be a virus. The genomes of viruses can be RNA or DNA, double-stranded RNA, single-stranded RNA, double-stranded DNA, single-stranded DNA. They can be linear or circular chromosomes, and they can have a single one or multiple ones. Here's an example of a herpes virus with a circular genome, circular chromosome, and consisting of 140,000 bases. The basic structure of a virus is the protein coat or the capsid and some nucleic acid inside them. The capsid, as you see, can come in various shapes, including the phage type virus, the ones that infect bacteria with that look like little spaceships. There are also these envelope viruses where the capsid and the nucleic acid are then surrounded by a membrane, and we'll see example of those, including influenza. Again, the phages are referred to as bacteriophages, and then you have some with a envelope, the membrane viruses. Okay, basic viral replication is that they need to infect a host cell, and once in there, in case of this virus, which is a DNA virus, you'll have transcription of the DNA to make some RNAs, and those RNAs will be used to assemble more of the proteins that are needed for capsid. The DNA will be replicated, and so then the replicated copies of the DNA and the proteins will be assembled into some new viruses that will then be shed from that cell. The cycle has been studied extensively in these phage viruses, the ones that infect bacteria. And so they have what's known as the lytic cycle, where they attach to the bacterium, inject their nucleic acid. The host's genome is disabled. The host cell is turned into a virus-making factory, making more copies of the DNA and of the proteins for the capsid, assembled, and then the cell splits or lyses. Thus the lytic cycle. To lyse means to split. Alright, now these phage viruses can also have what's called the lysogenic cycle, where after it inserts its DNA, instead of immediately taking over the host cell, that virus or phage DNA is incorporated into the host's DNA, and at that point we call it a prophage. The host cell can then go and divide on into two and other copies of the bacterium, and essentially the prophage is going to be copied each time as well, and so it can spread into a population of bacteria this way. Later on, it will then enter the lytic cycle, and the prophage, as you can see, separates itself. It will disable the host DNA, synthesize more of the nucleic acid and the proteins, and then lyse the cell. So this lysogenic cycle is essentially a dormant phase in which you have the prophage that's again incorporated into the host's genome. All right, this table 19.1, you don't have to memorize this by any means, but it's to show us examples of different kinds of viruses. Again, some double-stranded, single-stranded DNA ones, double-stranded RNA. Some of the names here should look familiar. Smallpox, herpes, cowpox. So SARS, that's one that was in the news a few years ago. Mumps, measles, rubella, these ones that we are commonly vaccinated against. Rabies, one that you see in mammals. And here, these retroviruses, which we're going to talk about in a moment. These single-stranded RNA viruses, HIV being a good example. So these viruses that have a viral envelope, what happens, and again, HIV is an example, influenza is an example, is that the envelope or membrane will fuse with the host cell, and the capsid and nucleic acid is released into the host cell. And in the case of HIV, it's what's known as a retrovirus. And so it has RNA, and that RNA is reverse transcribed into DNA using an enzyme called reverse transcriptase. That DNA that is made from the RNA is then incorporated into the host genome, one of its chromosomes. And at this point, we call it a provirus. You remember when this happens with phages, we call it a prophage, but here we call it a provirus. And it can remain dormant that way for a while, but then reemerge later, be transcribed, and then translated into the proteins that are going to be used to reassemble new viruses. And then when the viruses leave the cell, they take a bit of the membrane with them, and proteins that are specific to that virus. So, retroviruses, RNA viruses whose RNA can be used to make DNA, which then is incorporated in the host to form a provirus. Another example of a virus that becomes a provirus is chickenpox, which later, when it emerges, manifests itself as shingles. All right, so now viruses as pathogens. You're all familiar with vaccines. Vaccines are taken against viruses. Antibiotics are used against bacteria. Vaccines work when you take them before an infection, because then your immune system is much more primed to fight off the pathogen. It doesn't work so well if you already have it. Oh, I like this. Tastes good. I bet it did. All right, so I put this on here because you've probably heard this before. H1N1, H5N1. These are various versions of the flu, of influenza. And the H and the N refer to specific proteins that are in the envelope of this virus. And so the 1 refers to a particular type, and then there'll be other versions of the H's and the N's. Now, as pathogens, viruses can cause particular outbreaks. They can remain at relatively low levels, but then you can get spikes. And perhaps you're familiar, back shortly after World War II, there was what was known as the Spanish flu, an influenza pandemic that spread throughout the world and caused significant mortality in the United States and Europe in particular. Many thousands of people died from this infection. This was before vaccines were around. Polio was the first vaccine developed on a large scale. Also smallpox with Edward Jenner. He discovered that milkmaids in Europe who were exposed to cows and got cowpox were relatively immune to smallpox, and so that was actually the first vaccine. It was later in the 1900s that polio vaccine was developed. Animals aren't the only organisms that are infected with viruses. Plants can be infected with viruses. Some of the different colors you see in tulips are due to viruses. Now, even more simple than viruses are other types of infectious agents, these things known as viroids, which infect plants, and all they are is an infectious nucleic acid. They don't even have a protein coat. And this is one that apparently infects potatoes and causes them to have odd shapes. Other than having an odd shape, I don't think there's any other problems they cause. Now, in animals, you have these things called prions, which are these defective proteins that can be spread from one organism to another. The most famous example is mad cow disease. These prions, when they're in, they basically aggregate or collect in nerve tissue and begin to damage that nerve tissue such that the abilities of that organism are compromised and ultimately kills them. In people, this is known as Creutzfeldt-Jakob disease, and it was discovered in Indonesia, I believe, with these headhunting societies who would eat the brains of people they had vanquished, and the prions were transmitted that way. In cows, in the 1980s, there was an outbreak because the facilities that processed the cows took basically the waste material, the leftovers from the cow, which included parts of the nervous system, basically ground it up into a supplement that was then fed to other cows, and the prions were transmitted that way. Prions can't be removed by sterilization techniques because they're just these proteins that you really can't destroy. So basically that was pretty gross because the cows were eating parts of other cows and getting this disease transmitted that way, and then some people ate these infected cows and got the Creutzfeldt-Jakob disease. And so now there's much more restrictions on what you can feed cows, because when an outbreak occurs, it's a bad thing for the industry because then nobody wants to eat that meat, obviously. There's a lot of new emerging pathogens, particularly viruses, that sort of come from the wild and get into human populations, and in particular, as the human population has expanded and it's grown into more what at one time were remote areas, particularly in tropical areas, particularly in Africa. You get some of these diseases emerging. SARS, Ebola, well, SARS in Southeast Asia, Ebola in Africa, influenza in Southeast Asia, HIV in Africa. These started out in other animals and then through contact with people get spread into the human population. Now here's a figure that's in the back of the chapter. I want you to think about why we see these different growth patterns of the pathogen inside of people, why you get this relatively smooth curve in bacteria versus this sort of stair-step growth pattern in viruses. We'll talk about that in class. Okay.